I think the title of this series is Discovery Slam, and I've been racking my brain what kind of discoveries. I won't be able to tell you what the cause of autism is, and we haven't discovered a cure for autism, so if that's what you expect, this is the time to leave. In fact, well, to start off, these are the wonderful people in my lab who are doing all the work, and any mistakes and errors that I will present are going to be entirely their fault, of course. So I want to approach this from the perspective of Sir Karl Raimund Popper, who many years ago wrote that the way science advances is through conjectures and refutations. And refutations or falsifications are really important. And I want to make that point with regard to brain imaging on autism spectrum disorders, that it's sometimes important for advancing the field to understand why certain conceptions that we have or conjectures that people have made are wrong. First, brief introduction. What is autism? It's often in the media, so you probably know it has an increasing prevalence. And we diagnose it in terms of behavioral criteria that are in the socio-communicative domain. Behavioral is in bold. I'll tell you in a few minutes, I won't have more than a few minutes, why that is important. Well, actually, I'll tell you right away, because we can't look at brains and tell whether they have autism or not. One of these brains has autism. The one in the middle looks a little funky maybe, but it's the one on the right that looks perfectly normal. This is actually an axial, horizontal slice through the brain. We can't pinpoint where autism is and why a brain has autism. So we don't have any reliable biomarkers. Okay, let me give you a few, sorry, I'm jumping ahead. So autism is in the brain, but it doesn't seem to be anywhere in particular in the brain. On the other hand, it seems to be pretty much everywhere in general. One thing that people have agreed on in the past 20 years or so, that it's a disorder of brain connectivity. So it's not a localizing disorder, something to do with the wiring of the brain. So let me come to my refutations, and then I will explain to you why I think those are important and actually advances. One of them has to do with diffusion tensor imaging. That's an MRI technique. So it is a technique that looks at anatomical connectivity. And really what we're looking at is the diffusion of water in this technique. If we look at water in cerebrospinal fluid, it will diffuse isotropically the same in all directions. That's not very interesting. What is interesting is that when we look at water within an axonal fiber bundle in white matter, it will show anisotropy, or fractional anisotropy, that's the technical term, which will be up. It means it has directionality, it's an ellipsoid. And at the same time, overall diffusion will go down, and also the radial diffusion will go down. These are signs of a nicely organized fiber tract. That's what we're looking for. So we and other groups have done lots of studies using this technique, and this is just one example of many. And you see all these red spots. This again is an axial slice through the brain, roughly like this. And you see all these red spots on the green lines. Those are the tracts, and the red spots are the sites where we see evidence of white matter compromise in children with ASD through or per reduced fractional anisotropy, or increased mean diffusion, or increased radial diffusion, all signs of white matter compromise. And they seem to be almost everywhere. So that's an interesting finding. And many other groups have found that too. So this is from a review about 5 years ago of 48 DTI papers in autism, and they pretty much all found decreased fractional anisotropy and so on and so forth. So that's great. We have something. Problem is, only 10 of these studies reviewed there, and still the minority of DTI studies to the present day actually look at differences in head motion, and that's a big problem. So in my lab, Serafina Solders and Ruth Carper did a study looking at this, testing this empirically. So we are here only looking at anisotropy, remember this, which is a positive index of white matter integrity. And we're looking at it in a large sample, relatively large sample, and we see these spots of significant differences in autism. And then when we do quality control, they become a little less, but then when we match for motion, they go away. That's very depressing, very depressing. However, there's an upside. When we look at effect sizes, so not statistically thresholded anymore and not vulnerable to reduced sample size, we see that actually effect sizes are relatively stable in several parts of the brain. This is corpus callosum, this is another tract, superior longitudinal fasciculus. So, what does that mean? Well, it looks like a large portion of the autism DTI literature may have reported motion artifacts. But there are group effects. They are, however, much more subtle than previously thought. Why? I'll come back to that in a couple of minutes, if I'm still allowed to talk in a couple of minutes. I want to turn to my second refutation that is related to this, but for a more comprehensive picture. That one has to do with functional connectivity MRI. If you know anything about imaging and autism and connectivity, it's that the brain in autism is supposed to be underconnected. It's been in the press a lot, and a lot of people believe in this. This is a review 10 years ago, and these ideas are based on findings like this one. This is from a problem-solving fMRI task looking at activation in parietal lobes and frontal lobes, and they are very much coordinated in control participants and much less so in participants with ASD. Now there's a problem with this. We have an answer here, but what exactly is the question? Connectivity in fMRI is defined as correlation between signals or statistical dependence. So, for example, we have a stimulus or a series of stimuli, and they result in a time series that we detect in fMRI in a sensory region. And then this sensory region is connected to other higher processing cognitive areas, and they show correlated time series connectivity. But the problem is these two cognitive processing regions aren't actually connected. The correlations are driven by a task or sensory input. And this is a type of connectivity or signal correlation that is in fact often reduced in autism. That's been published in many studies. But it's questionable whether this is actually connectivity. There's a different kind of connectivity that has been studied and used much more frequently in the broader field, and that's called intrinsic connectivity. Sorry, now this is the only part where it gets maybe a little bit technical, but bear with me. You know that when we look at EEGs, we have different rhythms, from low rhythms, theta, alpha, beta, to gamma. You remember that from probably some biology classes or so way back then. And it turns out these are actually related to each other in the sense that lower frequencies, the phases of lower frequencies actually determine the amplitude of higher frequencies. And that goes down all the way to very, very low frequencies that we can actually detect in resting state fMRI. Don't make me nervous. So, and this is an example from a very first fMRI study. That's an activation pattern in motor cortex during finger tapping. This is something that looks exactly the same, but these are actually signal correlations during rest in the low frequency domain. That's called intrinsic functional connectivity MRI. It's considered to show us history of co-activation. And it is because brain regions talk to each other all the time, so even during rest, they show synchronized activity patterns. And it turns out that this gives us more insight into the functional architecture in autism. And in fact, the autistic brain is characterized by over-connectivity in intrinsic connectivity for many regions. This is an example from a study by Ina Fishman in the lab, and she shows that regions between a social cognition network and an action understanding network are talking too much to each other. And in fact, this crosstalk is associated with symptom severity. So, I've debunked a lot, I've refuted a lot. Do I believe in anything? I think the evidence that we have in autism is roughly consistent with a dual impairment model, in the sense that if we look at this cartoon network here of 3 regions, the networks tend to be less integrated in autism. So the connections are actually weaker. But the brain in autism also shows atypical connections with other brain networks, and that's reduced differentiation. And these are obviously reflections of impairments in typical mechanisms of network formation that we see in the healthy brain. Progressive ones like myelination, synaptic strengthening, that's 1 minute, right? Yeah. No. Regressive, oh God, I think I have one more slide. I have to present that one slide. Synaptic pruning, apoptosis, and so on. Because that's not good enough. Huh? What does all this mean? Why am I bothering you with this? Well, I've made it clear that if somebody tells you underconnectivity is what we know about autism, that you now know that it's a lot more complicated. The differences are more region-specific and subtle. Why are they subtle? Why do we find, for example, in my lab, with growing sample sizes, we tend to find less and less? Because, as I said, it's a behaviorally defined disorder. So if we consider ASD as a clinical label that from the biological perspective is actually almost a convenient fiction that encompasses many different biological subtypes, why would we expect to have the same or even similar findings in our groups of ASD? So the next step obviously is to look for subgroups. And I have another talk, 1.5 hours, but I'm not allowed to show it. So no, I don't, of course. We're working on that, and if Stanley invites me ever again, since I went over time, probably not, then I might be able 2 years from now so show you something about that. Thank you very much. Sorry to go over time.
Questions?
Mm-hmm. So you didn't promise answers, but as a scientist, you always have to promise questions. So what's the next step then? As you develop yours, it reminds me of kind of the conversation about cancer, right? At one point, cancer was a lumpy word, and now you have people thinking about it very differently. Where do you go next with this?
Well, I think it is, in fact, really important for us to leverage, for example, in vivo imaging techniques. Genetics, of course, it's been tried in genetics for easily over a decade, to find biological subtypes or variants of the disorder. Subtypes is already an assumption that they come in clusters. We don't actually know that, but we know there is a huge variability, not just behaviorally. That's easier to detect because we can interact and test these kids, but biologically and also in terms of their genetic risk factors. So, the genetics literature in ASD is kind of frustrating because way too much has been found. There are probably over 1,000 genes that have carry some risk for what we call idiopathic ASD that has not been explained biologically. We need to probably use imaging techniques to approach parcellation or divisions within the disorder to be able to even find a specific pattern of brain abnormalities that's then maybe treatable, or a pattern of genetic and environmental risk factors that would allow us to really go in and come up with some tailored, targeted treatments.
Axel, is it possible that the purported increase in ASD is due to the fact that more of the different syndromes are being defined as autism now than they were in the past? So it's not an actual increase, it's just better diagnosis.
Yeah, I wish that was the whole answer. That's certainly part of the answer. But I really have a distinct impression that there are risk factors that have increased beyond increased awareness and changing diagnostic criteria. I think it is truly an increasing public health problem.
Can I ask a question about that last question? And do you know of any studies that have looked at increase in diagnoses of ASD, probably children, in rural versus urban communities? Because my impression coming from a rural community is we used to have a lot more autism back then than we do now. Whereas living in the cities, we didn't call it autism, right? We call it the slow kid who didn't leave the farm.
Yeah.
But there was a lot of it, and that maybe I would be surprised. I would really like to see that study done, because I think there's less of
It's
that kind of
maybe
stuff happening in
done.
the rural communities than there was.
I'm not aware of that's something alarming. So I probably can't give you, but I don't have a good answer anyway. It's probably been looked at, but it's very difficult because awareness, as you say, back then might not have been. The terminology was different. Kid was maybe considered just whatever, what was mentally retarded, I think was maybe the word back then. Right?
One last question? All right, I'm going to ask one last. Okay, Bill.
You said earlier that there's no established biomarker. Do you mean there's none, or it's just not everyone agree
Well,
that
give me a
there's one?
definition of biomarker,
Yeah.
and then I'll tell you whether there's one or not. I mean, there's no single brain feature that occurs in all children or people with ASD and in no typically developing
Is
person.
that a way to use, for example, some disorders you have 3 or 4 kind of accepted biomarkers, and they can do a few different parallel studies plus chemical imbalance?
Yes, if you consider brain features that tend to, or in a statistical comparison, are more frequently observed in ASD than in typically developing kids, well, then there are thousands.
Okay.
So that is the problem. We don't know which. I mean, there are thousands because there are lots of studies that haven't been replicated. That's another. But I don't think we can start with rigor and reproducibility, because that's a big topic. That's not only a problem in autism, but it is definitely a big problem in autism research.
Thank you very much, Axel.
Okay, thanks.
Fantastic. Thank you.